All right. Hello, ladies and gents. So, we are going to go over cell communication today. And this part of biology is probably one of the more technical parts, but I also find it one of the most interesting parts at the exact same time. So, when cells talk to each other, they use a couple different communication methods. One of them is cell to cell contact. So when cells are physically touching each other, they can actually pass signals through each other's membranes. Local signaling. So if we have cell A and cell B a couple cells away, they can just bloop a little message over. Long distance signaling. So these are signals that are sent typically through lymph fluid in your body or the bloodstream or some sort of fluid like that typically is where these signals are sent. So when cells are sending communications from cell to cell, they do so through something called a junction. So a junction is where two cells actually physically connect to each other, and they connect the cytoplasm of adjacent cells through these. So we actually learned with plant cell walls, there's something called the plasmodesmata. So if you have two cell walls, there's little openings in it called plasmodesmatas, and between those little holes, the cytoplasms of those plant cells actually connect and they can share messages from one cell to the other. The other example that we have in here is in your heart, there are actually cardiac cells that are connected directly to other cardiac cells through little ports as well. And that's to make sure that they all contract at once so we don't get arrhythmia, which is where all the cells don't contract at the same time, which is one of the things that can actually cause a heart attack to happen. These can also connect cell to cell through receptors. So down at the bottom here, we actually have like immune system cells. So in your cells, you have those glycoproteins and glycolipids that we talked about in the actual surface of those cells. Those are your cell name tags. So immune cells will come along and they have a little, port. Your body cell has a little connector. They connect, and if it fits, it sticks, right? Then it stays in that body. So we can give and send information through these different things as well. Local signaling is a little bit different because we're not physically touching cell to cell. So, like if an immune cell is coming up and it's physically connecting to a cell, that would be cell to cell contact. Local signaling is when we have adjacent cells. So, we are going to talk specifically about the immune system next week and when we are sending chemical signals across. So, all of those like typical neurotransmitters, I'm sure you guys have heard of the words of dopamine, serotonin, norepinephrine, all of those different things get sent locally. So in the second picture here, we have an electrical signal traveling along the nerve that stimulates a release of some of these neurotransmitters. Those neurotransmitters go through a little opening called a synapse here. And they go through that opening and they touch the target cell, and then on that target cell, there's little receptors that will accept those little chemicals that we call ligands or ligands. I've heard it both ways. Accept that to cause some sort of a conformation change within that cell. Here, we also have paracrine signaling. So this is when we have a cell that secretes things. So once again, your rough ER, your smooth ER, all of these things in your cells that physically make something get sent to the Golgi body or the Golgi apparatus. They get packaged into a vesicle, and your cell secretes them from those vesicles that are made from the Golgi apparatus. So they get secreted. When those proteins or lipids or whatever get made in your cell get secreted, they have target cells with acceptors that will accept that molecule, then causing some sort of conformational change or some shape change or some sort of a domino effect signal to happen. Specifically, scientists like to use the example of yeast sexual reproduction for identifying mates. Because if you can imagine, if you're a little wow, if you are a little yeast, you don't have yeast, dating apps to find a yeast that you want to mate with. So they have exchange of what they call mating factors. So we have a male yeast or a yeast of mating type A and then a yeast of mating type alpha. So we have like a typical male and a typical female. One will release an alpha factor that gets recepted onto the A-type cell. The A-type cell will release an alpha factor that connects with the alpha cell, and then they connect together and fall in love, and then they have a new A alpha cell. These A alpha cells are now a zygote, and then includes all the genes from the A and the alpha. So we can think of the A and the alpha like a sperm and an egg. And then they connect, and then they make a baby yeast that can make bread, which is delicious. So the next one we have would be long-term or long distance, not long-term, long distance signaling. And this is the use of hormones. So plants and animals actually use hormones. It's weird to think of plants using hormones, but they do. They have different growth factors that are released at different times in their life or different stages in order to grow in certain ways. So growing taller, growing wider, anything like that. So when we're thinking about hormones with animals, we think of things like testosterone, estrogen, insulin is also a hormone, and they can affect so many different things. So your pancreas, which is where your insulin is made, is like right here, but you need insulin all over your blood. So that insulin molecule is going to travel in your bloodstream all throughout your body, even though it's only made in your pancreas. So same thing with like your pituitary gland, that's like your puberty part of your brain. That can make certain hormones that get sent all over your body through your blood cells, or not your blood cells, oh my gosh, your bloodstream. And these can be either protein hormones or they can be a steroid type cholesterol based hormones as well. So how does all of this work? It is so stinking cool and crazy and complicated all at the same time. And we call this pathway of cells communicating with each other signal transduction. Okay, signal transduction. So we are going to convert signals on a cell's surface into some sort of a response. So we have a cell, something touches the outside of that cell, and then it makes some sort of a response inside. So it's either going to make something, it's going to turn a gene on, turn a gene off. And the really cool thing about this as well is that mammals, plants, and microorganisms like bacteria, amoebas, they all have like extremely similar / the same cell signaling pathways. So that probably means that this signaling and this way of communicating for cells had a really, really early origin. This is probably like the most important part for you guys to know, the 3 phases. We have, look how fancy I'm getting, reception. So this is when we have a signal molecule over here, typically called, signal molecule that's going to attach to a protein on the outside of that cell. So we know from enzymes when we have a substrate in an active site that causes some sort of a conformational change to that protein that's going to activate or break down or build up or whatever that substrate. Here it's going to be similar, but these signal molecules, we are going to call them ligands or ligands. When it attaches to that protein is going to change shape. So think of it like, oh my goodness, think of it like a domino effect. So that's going to start the dominoes. Step 2, right up there, is transduction. So there are relay molecules. You don't just have the first domino, you got to knock them all down in a line. So we have a bunch of different things that have to happen. And this is a safety check for us because if there was only one thing that had to happen, it'd be really, really easy to accidentally trip that switch or turn things on and off. So it's kind of a safety mechanism for ourselves. And then lastly, look how technologically advanced I am. We have our response. So this is the activation of some sort of cellular response. So it can be making a product, stopping making a product, turning on a gene, turning off a gene. Lots of different things can be the response. So we have reception, transduction, and a response. So once again, reception. This is when we have a signal molecule called a ligand binding to a receptor protein. So the ligand and the receptor are going to have a very unique binding where they're very, very specific. If you don't have a receptor or if you don't have a ligand, you're not going to get binding. So a lot of mental neurological disorders are from poorly shaped protein receptors or poorly shaped ligands that don't fit, that don't initiate that whole response. Step 2 is going to be our transduction. So the signal hits the cell, hits that protein, we have some sort of a conformational or a shape change, and then that turns into a cellular response. So we call this transduction, sometimes a signal cascade, where we have a series of events that are going to happen to get to our final result. And this is going to be a multi-step pathway, and it also helps to amplify the signal. So think of it like a pyramid scheme, right? You have one person up on top, and then you have all the other little people down below. So it just takes one person to start all of the stuff down below. We're like a chain letter, right? When one person gets it, all of the people figure it out down below. So multi-step pathways can help amplify the signal, and it's a safety step for our cells. We also have something that's called a secondary messenger. So if we think about our cell membrane, so this is our cell membrane, we have our ligands coming down. So my watch, think of that, my watch as a protein. So the ligand comes down, attaches to that protein, some sort of a conformational change. So we have a conformational change down here, and then that is going to cause some sort of response. The ligand never went inside of the cell. So we have to have secondary messengers that are activated by that shape change down below. Okay, we have to have secondary messengers that are activated by that shape change down below. Step 3 is going to be our response. So this signaling leads to regulation of cytoplasmic activities. So these are things that happen in the cytoplasm. So think activating the rough ER to make something smooth ER, something like that, or transcription. So remember, transcription is when we are activating genes, turning them on or turning them off, saying, hey, you should probably start making that thing, or hey, you should probably stop making that thing. Lots of different things can happen. Oops. Okay, so this is a phosphorylation cascade. It is crazy. Take a minute, pause the video, kind of read through everything on here. But basically, what a phosphorylation cascade is we have our signal molecule, attaches to our protein. The protein changes shape. Stuff happens inside. This is the stuff that happens. So up here, we have an inactive protein kinase. Kinase, kinetic, means to move. ACE means an enzyme. So we have an inactive moving enzyme, okay? So here we have some sort of a signal molecule. This is the first domino that fell. That domino attaches to that protein kinase. Now we got a substrate attaching to an enzyme. Think about it that way. Activation, okay? What happens here is we have another inactive protein kinase. And here we have ATP. So what happens is ATP will attach to this whole substance and become ADP. ATP has 3 phosphates. ADP has 2. We can see that extra phosphate attached to that other protein kinase to activate it. And this happens over and over until we get to what our final product has to be. So phosphorylation is when ATP donates a phosphate in order to activate some sort of a molecule. Because remember, phosphate as a functional group is very, very polar. So in order to activate it, it causes this whole chain of events. So the benefits of a secondary messenger system are amplification. We have one signal molecule hitting our cells and all of this stuff happening inside, and we get lots and lots and lots of product. Lots of amplification. It cascades and we get a fast response. So the benefit of how this system works is we have one signal, receptor, big, big, big response. So these pathways can regulate and turn on and off genes. Okay, they can regulate and turn on and off genes. So we have reception, We have our ligand, receptor, phosphorylation cascade. It can activate some sort of a transcription factor that can turn on or off a certain gene. So it can either activate making mRNA or it can deactivate making that mRNA. For us, these types of receptors are not going to be super important, but you might see them in certain readings. So know that there are three main types of plasma membrane receptors: a G protein, a tyrosine kinase, and an ion channel. The two most important ones that we're going to talk about are going to be our G proteins and our ion channels. Ion channels are specifically with nerves. We're going to talk quite a bit about those. G proteins are the example that I gave already. Tyrosine kinase is a multiple receptor pathway. We're not really going to hit on that so much, so don't really worry about it. The G protein is the most common type, so we will be talking about this one. It's very common, and this results in a single response of something. So basically how this works is we have our G protein, and here we have our inactive G protein inside. The receptor links, this G protein gets activated, it goes over to activate the enzyme to do some sort of a cellular response. Okay, that's all it is. This is our secondary messenger inside. Whereas the difference with a tyrosine kinase receptor is that here we have a single molecule and a binding site, or a signal molecule, and a binding site on top of our tyrosines. These will come together into a dimer. So when we have two of them, it's like Wonder Twin powers. They get their ligands, they connect and become activated. And now if you're looking at all of this stuff down here, we have lots of different places where molecules can attach to begin a phosphorylation cascade. And we can actually get relay proteins doing two separate cellular responses from one signal molecule. So that's the difference. So if we flip back G protein, one ligand, one response. Tyrosine kinases, one ligand, two or more responses. Ion channels are used quite a bit, I'll go down here, for neurotransmission. So we're going to talk about that with the nervous system. They are also used quite a bit for certain signalings to get differences in charge on either side of a membrane. So those are going to be really important later when we talk about certain other cellular processes. So when ligands bind, it's going to turn on or off a channel. So ligand's going to bind. What happens? So here the protein is closed, here the protein is open, and now these ions can go inside. So ions are either negative or positively charged. So it's going to create a net difference of charge across that membrane that's going to cause some cellular response. Oops. We do have one little difference with some of our molecules too. If all of that wasn't confusing enough, we do have one difference with this one. We also have intracellular receptors. This is where your target protein has to already be inside of the cell. Therefore, your ligand has to be hydrophobic. So why can a signal molecule meet its target inside of the cell? Well, if it is hydrophobic like testosterone, that is a steroid that is made of cholesterol that we know is a lipid. This is why biochem is important. We know that lipids are hydrophobic. That is going to be able to go through a membrane very easily. So it does not need an extracellular receptor on the membrane. The receptor can actually be inside because the ligand itself can actually go inside of that cell. The significance of this is that through millennia, we think that this is how cells actually talk to each other. And bacteria actually secrete molecules so they can sense how big their own population is. So we are going to watch this video on quorum sensing. It's a TED Talk. It's about 20 minutes long, and it is by far one of my favorite TED Talks ever. And it does a really good job of talking about how these cell signaling ideas are actually used in bacteria. And this is where we've actually studied most of what we know as scientists about cell signaling. So I hope this video helps to go over the notes and clarify them a little bit.